Welcome. Dr. John here for extraktTalks. I'm joined here with Stefan Egan. How you doing, buddy?
Fantastic. Yourself?
Another great day to be alive.
Every day's a blessing.
Yes. Every day is a great day at extraktLAB.
I would agree with that. You get to work in an industry that's changing the world. You can't be mad at that.
Yeah, so we're going to talk about one topic that obviously is really important for pharmaceutical formulation, and that is water solubility. The rec market's way ahead of the pharma guys on this one.
I mean, that tends to be the case, though.
In terms of the science, there are all of the different ways to get oil into water and then into your body.
Right.
That's kind of what they're all looking to accomplish, and they're looking to accomplish that with stability, so that, and then they can stick it on the shelf for 2 years and you can still take it. It still has the potency.
It's not sticking to the sides of the can or dropping out. Yeah.
Making sure it's not toxic too. That's the other thing. In a lot of ways, the states people are, experimenting with a lot of these water soluble items, and they're using, emulsifiers, I guess, right?
I think that's pretty much the standard right now is emulsifiers, right?
Yeah.
Sonication, emulsifiers.
Yeah, so you use sonication, you get an emulsifier, make little particles, and it tends to make something, it encapsulates it, I guess, if you will.
Puts a blanket around the oil.
Yeah, puts a blanket around the oil, and then those particles, they're put onto or into some sort of powder, or maybe you just put them directly into your tincture
or beverage or
beverage or whatever it is. So it's kind of interesting. There's lots of different ways to do that. We're going to talk a little bit about what water solubility is. One of the main methods to do that would be to make liposomes from sonication.
Right.
We're going to talk just slightly about bioavailability, so let's kind of get to it. So let's talk about water solubility. I mean, let's talk science first.
I think at the core level, it's just how much can the water hold?
Yeah.
Right?
Right.
How much can we ensure that this water is holding before it separates out of the water and becomes two separate layers, right?
Exactly. So yeah, I mean, it's usually you have a certain mass of water. So I think that the water solubility numbers are standardized, so they're reported on like 100 grams of water.
Right.
So 100 grams of water, so the maximum amount that you can dissolve in 100 grams of water is your water solubility.
Right.
And obviously you have things like sparingly soluble, soluble, and then not soluble. So hexane and water, they don't mix. Oil and water, they don't mix, right? And a lot of that has to do with like polarity and the polarity of the molecule. You don't really have to get into too many details, but you have surface tension, essentially. The surface tension of water is really high compared to the surface tension of, say, ethanol or even the surface tension of, say, a nonpolar solvent like
and
hexane or butane.
What's the significance of the surface tension? So in my mind, it's how long is it going to stay encapsulated or water soluble, right? If there's too much surface tension, it's going to eventually separate, right?
Well, okay, I think it goes really back to the fundamentals of the thermodynamics of solubilization, okay? You think of like surface tension, if you have like water on a stainless steel surface, it automatically spreads out, right? That's because the surface tension of the water and the stainless steel are similar. They're very high surface tensions, okay? If you take that same droplet of water and you put it on, say, a piece of PTFE or something like a Teflon,
right?
Okay, you'll see it bead up, right? And the reason that is because the surface tension differences are really high, so it's not wetting,
right?
And so you can think of like water solubility as following that process. That's a thermodynamic process where they have concepts like free energy, where the free energy is just as favorable for it to wet the surface, whereas opposed to something that's so far away in surface tension, it doesn't wet the surface, so therefore it's not soluble.
Right.
Kind of the organic chemist's way of thinking of this is a little different than the thermodynamic chemist's way of thinking of it. They think of it primarily in terms of polarity,
right?
Like a nonpolar solvent, right? Like butane is a nonpolar solvent, right? Propane is another problem. We got hexane,
good old
heptane,
hydrocarbons,
nonane, dodecane, all those guys.
Yeah,
I don't know. The polarity is a really big thing there. So obviously the water is pretty polar.
Right.
So that's how I think about it. Surface tension is the thermodynamic way of thinking of it, maybe the more the chemical way. And then the polarity, you're really making arguments based on electron density and polar molecules versus non nonpolar molecules and things like that. So I think there are different models of solubility.
Right.
So,
Which just goes to show how complex this actually can be.
it
right?
can be, yeah. If you're in the pharmaceutical world, you are in command of all of that knowledge, right? Whereas maybe a little bit less so in the more consumer world, I think, and
Absolutely.
just trying to figure out what works. If you have command of the basics, you can manipulate the particulars so that they're always consistent all the time. Some of the variability that you might see in the consumer market would be particle size, large particle size distribution, for example, maybe excess oil versus the amount of emulsifier, maybe too much emulsifier.
Yeah. I see that one a lot.
Yeah. Well, that's an issue because that's where toxicity comes in.
For sure.
And different emulsifiers have different toxicity levels. You got the old tween.
The
you
tween
have
20,
lecithin.
40, 60, 80, yeah, all of them.
Yeah, exactly. Eat that stuff, and you'll find out that's not very good. You see, you have some of the like oil and water or water and oil emulsions, like they'll taste like soap.
Yep.
So
Plasticky soap.
plasticky soap, yeah.
But it's weird because they're all typically labeled as generally regarded as safe, right? So we as consumers think, well, we can just eat this stuff in abundance.
Well, yeah, here we go.
And not have a problem with it.
Get that palm olive oil, suck on it, suck on the bottle. I think, I guess, there are toxicity levels. There are limits to that, right?
Right.
I think they're generally regarded as safe in certain quantities or, within certain uses. But, like, for example, if you can dive from water, even though it's generally regarded as safe.
Right. Fair point.
Yeah, so that's what water solubility is. There's all kinds of ways to manipulate it. I mean, you've got a lot of experience with that. You had mentioned making liposomes. And
So we would just typically take long chain triglyceride blends and blend them together to allow us to use sonication to encapsulate the oil and give it the appearance of being water soluble. Right. So in my experience, nothing that I have done is actually water soluble, right? It just appears to be transparent, and it's not like we're taking an oil molecule and water molecule and making them one. Right. That's just not at all what we're doing.
Right. So you're not like encapsulating typically, or you're sonicating, right?
It's use sonication. Yep.
Okay. So take us through that process if you could, step by step.
So I am warming up my oil and I'm warming up my long chain triglyceride blend or lipid blend, and so they're of equal temperature, so they can blend and homogenize more effectively. And then I am using a small amount of water with sonication, gradually increasing the hertz of that sonication to ensure I'm breaking down those particles
into smaller
size. Exactly. Which then leads into bioavailability. Right? The smaller you can get those particles, the more effective and more it's going to work.
Right. So you have so there's obviously going to be a large particle size distribution that comes out of that, right? So probably some of them, like if you have, if you have maybe some really, really small particles, then you have your average, and then you have your really large particles, right? So I guess the trick is to get that particle size distribution really tight, right?
Right.
Do you have an idea how big typically you would get in? Yeah.
So I believe it's around 40 micron.
Oh, wow.
Microns is the goal.
That's pretty
big.
That is really big, actually. So you get like a lipid bilayer, or you can have a single liposome, Yeah, so then you shake it up. There's a critical micelle limit, but I don't know. I don't think that has anything to do with this. You can do it. You're using mechanical agitation or high shear to get it into the liposomes.
Right.
So, okay. And
the goal
is
to not have to shake it up at the end. That's the goal.
Yeah, exactly.
We want to make sure it stays soluble within the water or the beverage or the tincture, right, without having to shake it up or give it that shelf stability we're looking for.
Right. What kind of shelf lives do you typically get out of what you do? So
I personally have done 9 months. And that's as long as we've left it on the shelf without any crash out or precipitation from the solution. I'm assuming it can go longer based on it not crashing in that 9-month period, but I haven't gone beyond that.
What about the stability? Have you seen degradation come out of that? Or
no. So as long as it's stored in proper cannabinoid type storages, not direct UV light, ambient temperature, dark, cool place, traditionally, it should work just fine. You shouldn't see any degradation until that 12 to 14 month timeline. And that's when you start noticing things starting to fall off a little bit.
Right. Okay. So typically what you would do is you would take a placebo, right? You'd make liposomes without any stuff in it. And then that would be your blank. And then you could, you could add your drug in, and then you would come up with a formula. Right. You put that into the finished good bottle, all wrapped, labeled, everything, and then you'd take a sampling of those according to the sampling schedule, and then you'd put those into a shake and bake oven, basically, controlled humidity, controlled temperature, and you do that over time. You do it at a couple different temperatures, like 25 degrees Celsius and then 40 degrees Celsius or something like that. And so you'd have a double, and then you'd be able to understand what the effective temperature is, and also just conditions. And then you typically would test the CBD, THC, or THCV or combination.
Right.
You'd test that over time, and you would actually start to see small amounts, but I think, basically 10%, the number that the ICH gives is like 10%. Within 10% of the label claim, it's okay. And it takes a certain amount of time to get there. What you're saying is that's pretty constant over that amount of time, like 9 months is what you were looking at. Yeah, and so that's what you would do for a stability test if you were going to do a stability test. And I know a lot of people have looked at that. The other thing would be, like, you need to understand what the degradation compounds are, right, in that case. So you have CBN would be a degradation compound, right?
I think that's ultimately where it all goes, is the CBN. But you would slowly start to see a decrease in each of the other molecules and an increase in CBN, because that's where the path leads to.
So there's your stability indicating that's your impurity profile. And as long as your excipient doesn't degrade, that's the other thing, right?
We want to make sure we're using
and what does it degrade to? So there might be other peaks that come up in the chromatogram afterwards. So now we just kind of went down an incursion. This is free association here, but this is all about water solubility. So these are some things you guys got to know and think about when you're doing your water soluble testing. And if you're going to try to make liposomes, give us a call. Certainly we can help you come up with a method and figure it out. I mean, there's so many methods out on the internet right now.
Tons and tons. Yep, absolutely. Yeah. That's the new encyclopedia for the cannabis operator, is just the internet. Right.
And there's just going to be so much information there that's not such a good idea. By the way, just one note on that is, and going back to the whole idea of toxicity, you can really hurt yourself actually by doing the wrong thing. That's something to keep in mind when you are doing your formulation. You just want to make sure that you have a really good idea of the concentration levels and that they're within the limits so that you're not actually making a toxic compound for the people that you're dealing with.
And if we're talking cannabinoids, make sure we're not making it over potent as well, because as we reduce that particle size, it becomes more effective.
Yeah.
And if you tell someone that's 10 milligrams, but it's at a 20 nanometer, like, it's going to send them to the moon.
Isn't that interesting? Like the 10 milligram limit or whatever for like beverages or whatever, that's all predicated on not extremely bioavailable because of the timing, that came out 10 years ago?
Yep, of like a 30% uptake.
Yeah, now it's quite good.
90 to 100% if you're hitting the right size. Like, it's a whole different experience at a much lower
and your
level.
onset time should also be
rapidly
faster. Yeah, so we didn't really get into that, but that's another benefit of the water soluble side of it, right?
Yep. Which is why it's more of a medicinal application. I feel like it gives that rapid onset for quick relief, if that's what you're looking for.
Right.
There's definitely benefits to it if you do it right.
Right. All right. So sounds good. Good talking to you, man.
Appreciate the time.
Yeah. Talk to you later.
Take it easy.
Thanks for everything. Bye now.